You can fail as fast as you want, as many times as you want. Failing does not necessarily mean that you're going to figure out how to do things. It just means that you're going to keep failing, right? I'm not interested in failing faster. I'm interested in being right as fast as possible, as early as possible. I don't want to fail 1,000 times before I get to the right answer. I want to get to the right answer within 10 tries.
Guillermo Vela is the CEO and co-founder of NeuScience, a VC-backed TechBio company using AI to discover novel cancer biomarkers. He went viral, at least in the TechBio scene, with his 5 TechBio myths that must go article, in which he explained why it doesn't take $2.5 billion to develop a new drug, and why technologies like AI won't make drug discovery faster, better, or cheaper. We discussed that article, we discussed Guillermo's contrarian view of cancer, we discussed why we're doing more science than ever but not seeing results, and how his ADD brain is a superpower. I hope you enjoy. The thing I wanted to talk about from the article was this myth you tackle that it takes $2.5 billion to develop a new drug. And you suggest that it's probably closer to $100 million. But then you give this really interesting bit of context, which is of Eroom's law. And to understand that, the context you need to know is about Moore's law. So that's a law in computer science that basically the number of transistors in a circuit doubles every 2 years. And my kind of layman understanding of that is that every 2 years, computers get about twice as fast, and it's an exponential curve. And Eroom's law is the pharma equivalent, but actually it's showing that we're actually becoming less efficient. So we're spending more money trying to develop drugs, but getting less useful output, which doesn't really make sense. So can you square this circle where you're saying that it doesn't actually take as much as we think it does to develop a new drug, but at the same time, we're getting a lot less efficient at it? Like, this just doesn't make sense to me.
So, by the way, Eroom's law, like the reason why Jack Scannell et al. Named it that is it's Moore's law backwards. So it's a clever kind of punning name for it, where he's saying, look, you have the technical version of this, which is Moore's law, that exponential advancement in technology. And then in pharma, we have the exponential, deceleration, if you will. Like, so he called it Eroom's law, which was clever. And I think one of the reasons why it was a sort of resounding hit, but also because of the implications of the observation to your question exactly. Why is our efficiency decreasing when our technology is increasing, right? Like usually efficiencies increase with increasing technology and costs decrease with increasing efficiency and increasing technology, right? I mean, people talk about this in the pharma industry a lot, where it used to take, I think the first genome cost a total of like $2 billion to sequence the first genome. And today, I don't know what the going rate is, but I think you can get it done for like 100 or $200. So, that's Moore's law in action. Eroom's law is that we have all this genetic information and we have no freaking idea what to do with it, right? And I kind of allude to this a little bit in the article, where I say maybe some of our decreases in efficiency are actually due to our over reliance on technology and sort of outsourcing the critical thinking to technology. So, a lot of times technologies also bias us towards the way of doing things. Like if you think about it, like how did those like OG scientists from the 50s generate so many like discoveries, right? They just had a freaking microscope and the power of observation, and it's just fantastic. Like they were just legit scientists and legit thinkers and sort of philosophers in their own way. For example, the molecular revolution that came at the heels of us being able to sequence, I think led us terribly astray in cancer, because there was a compelling narrative that we could sell to grant agencies, to investors, to anyone that would listen. That was that because we can now decode the genome, we can decode cancer. It's as simple as finding out what genes are malfunctioning and then addressing the problem. And so we took a very genetic, gene-centric view of cancer. It was like a gene horoscope of cancer. And we've been humbled since to know that biology is far more complicated. And there's what's called emergentism or holism, which is basically what it says is that the sum of the parts are not equal to the individual parts, or the whole is not equal to the sum of the individual parts. Like A plus B does not equal C, or like 2 plus 2 does not equal 4. It's more like 2 plus 2 equals like 5, right? Where you're like, how do we go from here to here? And an example of that would be like consciousness, right? We know consciousness resides within the brain somewhere, but where? right? And you'll never understand consciousness by studying the functions of a neuron, right? The neuron is what enables consciousness, but it tells you nothing about how consciousness works or where it even resides, right? You could dissociate me and put me right back together. And it's like, well, when you dissociated me, where was consciousness, right? What level, what number of neurons do we need to hit consciousness, right? No one can answer those questions yet. Consciousness is an emergent property of biology where something much more sophisticated emerges that you can't predict from looking at the individual parts.
So we've almost had too much of like a reductionist attitude towards these problems where we've just thought that we understand or we can now look at genes in more detail. So they must be the explanation for everything. Is that kind of where you're heading?
Yeah, absolutely. Absolutely. So maybe this is a good point to talk about my contrarian view of cancer. Before you and I were talking, and I mentioned how, my very Mexican sense of humor has had a big influence on my life. And this is a classic example of how a stupid joke that I made in the lab, because of my very Mexican sense of humor, of always trying to look at like some joke that I can make from something, or look at the irony of something, or make a pun out of it. Kind of completely ended up transforming my perspective of cancer. At the time, I was being taught how to culture brain cancer stem cells, and these are the most aggressive, like, cancer cells you can imagine. They're resistant to chemotherapy and resistant to radiation, and we can't kill them with anything, right? You can nuke them and you won't kill them. I mean, they're just incredibly robust. And as they were teaching me how to culture them, they say, well, whatever you do though, don't expose them, don't add serum media because they will stop growing. So, and that's kind of like a big thing because most cells are grown in serum media, and serum just has a bunch of stuff that's in the blood, like dwarf factors and a bunch of other stuff that helps cells grow in culture. With these cells, it's like whatever you do not give them serum media because they will stop growing. And so my very Mexican sense of humor, I'm like, oh, well, guys, guess what? I just found the cure for brain cancer. It's serum media. Right. And the stupidity of the joke was that obviously, like, serum media, like, brain tumors are, exposed to elements of that already. But that, it was kind of one of those where my friend, mentor at the time, she kind of gave me like a pity laugh, and then we all just kind of scratched our heads and like, anyways, moving on. But it stuck with me. I was like, wait a minute, like there's something here that's not quite making sense. Are you telling me that these cells that we can kill with anything, we make them stop with serum media? And actually, like, a dirty secret, if we can call it that in science, is that it's actually quite easy to make cancer cells stop growing in a lab or in a dish. And that gets me back to the point I was about to make, which is if I asked you to define cancer, or, if you asked me to define cancer according to consensus, I would say that we define cancer as a disease, or if you want to be more specific, more correct, a collection of diseases that are characterized by the uncontrolled proliferation of abnormal cells. And what makes them abnormal? Presumably because they have genetic mutations. Except if you actually think about it, no part of that statement, or like no key assumptions, no one assumption in that statement is actually correct. Every part of that statement is incorrect. And that may sound shocking and almost insulting because, like, wait, that doesn't make any sense. We all know that that's exactly what it is. So many brilliant people work on it. They all agree, but it's actually very easy to prove. So uncontrolled proliferation, I just gave you an example. Well, clearly it's controllable, right? And don't take my word for it. In the clinic, we see it all the time. It's called dormancy, right? Tumors can enter dormancy and then exit dormancy maybe some months later, maybe 10 years later, right? So clearly cancer cells are very capable of controlling their proliferation, which has profound implications for the idea of driver mutations, because clearly these tumor cells have these quote unquote driver mutations, but they're not, they can control their proliferation. So it's not a purely intrinsically driven process. It's clearly a conversation with the extrinsic environment, microenvironment. That there's something there that is promoting or inhibiting the behavior of these cells. And so, from that observation, I start questioning, well, the premise of like, well, are genes really drivers? And my, simplistic answer, rudimentary answer to this, they're not drivers, they're enablers, right? And cancer is sort of like the perfect storm of a bunch of fail-safe systems that fail, essentially. But there's no one thing. And okay, applying the scientific method again, People that are born with a susceptibility to breast cancer because they have a BRCA mutation or colon cancer, it increases your chances of getting cancer, but it doesn't predetermine you to getting cancer. So again, if you think about it scientifically, it's like, we're trying to prove and disprove. We think it's the cause because we see a correlation. But the opposite would be, well, if you have this mutation, does that mean that you always get cancer? And the answer is a resounding no. So clearly that in itself is not enough to develop cancer, right? Another perfect example, people with Leaf-Dominic syndrome are born with a mutation in P53, which is classically known as the guardian of the genome and involved in helping cells repair themselves. People born with Leaf-Dominic syndrome are heavily predisposed to getting cancer. And most of them actually do later in life, but they don't get cancer everywhere, right? Or there's no random assortment of cancers that you see manifest in the clinic. It's very specific to certain tissues. Why would that be when that mutation is present in every single cell in their body? Why aren't they getting cancer in their big toe or their heart or their ear, right? It's very specific to the context. And so clearly there's something there that we're still not fully identifying that goes beyond just guardian of the genome role, right? And so anyways, so clearly it's not a disease of uncontrolled proliferation. Well, what about abnormal cells and mutations? I'll just explain to you why. Mutations in and of themselves don't explain cancer. And then more importantly, cancer isn't even a disease of cells. There's no such thing as, philosophically speaking, as a cancer cell. And I think that because cancer isn't a single cell disease. There's a reason why bacteria don't get cancer. Have you ever heard of a bacterium getting a mutation and being like, oh, I can't stop dividing anymore. I'm just proliferating indefinitely. Of course not. Cancer is a multicellular disease and it can only manifest, it can only arise at the tissue level. It does not exist at the genomic level. It does not exist at the cellular level. It exists entirely at that tissue level, which is why we've always relied on pathologists to diagnose cancer. And in fact, pathologists wouldn't describe cancer as, I would think, as a disease of proliferation of normal cells. Certainly they look at things like Ki-67, which is a marker for proliferation, but a high Ki-67 score is around 20%, meaning that 1 out of 5 cells within the tumor appear to be dividing at any one time. 20% of cells. That to me does not seem like uncontrolled proliferation. That is clearly not exponential proliferation, right? So if you think about all these things, it makes you kind of reassess how we're going about cancer, right? Why are we trying to stop proliferation? Well, the answer is for a historical reason. In the lab, it's just easier to, what are you trying to do? Well, we know cancer is a thing that grows. And if we do this, it stops growing in the lab. Well, that was kind of one of the few things that we could measure objectively in the lab in the field, right? And that's kind of why that stuck around. But if it's not a disease of proliferation, then why are we trying to stop proliferation? I'm not saying we shouldn't. I'm just saying, let's think about it critically. Is that really the right readout? So my sort of controversial or contrarian view on this is that actually the cancer models that we have today are pretty good. Some of them, not all of them. Some of them are abysmal. But primary cell lines, freshly created from tumor tissue from the lab, have a high concordance with clinical responses. That's not the same thing as durable responses, but they're at least pretty highly correlated to how the patient will initially respond to the treatment. And that makes sense because there's this sort of epigenetic memory in the cells that represents sort of the environment that they were in. And over time, they start losing that memory or they start rewriting their memory. In some ways, what I was getting to is molecular revolution led us astray because it made us gene-centric. And it was a sexy, pattern to sell. Suddenly things seem feasible and simple. And it was far from that. But it's just as important for us to be critical about the underlying assumptions. So I'll give you another example. Alzheimer's, right? I don't know if you recently learned, or heard about the news that the scandal, apparently the data that led the world to pursue the beta amyloid hypothesis was fabricated. And obviously I can't speak for the companies in the space. And they may have a perfectly reasonable answer for this. But I wonder, why didn't all the AI companies in Euro find that, right? Maybe it's because, a perfectly reasonable answer, and it might very well be the answer, is that they actually weren't pursuing that question. They weren't really interested in pursuing that avenue of research. So they actually never looked into it. But another reason might be that they did look at it and they just never noticed because the data that we're feeding our algorithms was inherently biased to believe that that was actually the underlying hypothesis, or that the underlying hypothesis was correct. And so that's an example of how AI won't get you out of a bind if your underlying assumptions are incorrect.
That's so, so fascinating. And I think what you've said about cancer and what you've mentioned about the Alzheimer's scandal fits really neatly into this concept I came across a few months ago of the half-life of knowledge. So typically we use half-life to describe the time it takes for a drug to halve in concentration once it's in the body, effectively how long it takes to decay. There's also a half-life of scientific knowledge, and it varies across different fields. The things I've seen quoted are that in some sciences which can be less rigorous at times, like psychology, it's about 7 years. So that's the time it takes for our knowledge to halve. And in medicine, I found one paper on it. It seemed to be about 40 years. So it takes 40 years for our knowledge of medicine, for half of it to be shown to be incorrect and for us to make new assumptions. So it's super fascinating that you're thinking of these ways in which our underlying assumptions might be incorrect, because no doubt it will inevitably come true. The other part I thought that was super interesting about your article was the assumption that tech bio will make drug discovery cheaper, faster, and better. The figure I always see quoted is the time it takes from a discovery to get from bench to bedside. And the number I've seen is 17 years. So that's how long it takes from once the discovery is made in the lab to actually helping patients in the real world. Feel free to correct me here. My understanding is that TechBio focuses on the time frame before that, so before the discovery is made. And was your kind of point that shortening that or making that period more efficient won't actually have a massive real world impact? Is that correct? Like, feel free to say, no, that's not what I'm saying, but like, what were you trying to say there?
Yeah. Yeah, you're mostly correct. So I think there's just some clarifications that weren't necessarily discussed in the article. So they're worth bringing up now. First, let me start by saying that, the idea that it takes 17 years to develop a drug, I mean, it's potentially technically true, but also kind of irrelevant, because a lot of those years are already baked in today, right? Meaning that if you give me $100, $100, $100 million, I don't want to say very easily, but quite feasibly, get a drug approval in 5 years' time. Why? Because there's already, all this baked in research that's been going on over the past decades that I can simply in-license and then try to, push through clinical trials and potentially get an approval, right? And so if I start a company today, it's not going to take me 17 years to develop a drug, right? And then that's kind of the cheap trick that a lot of us in the tech bio field kind of implement, which is that no one is really going after novel drug targets, right? I mean, this isn't to boost our own image here. I'm quite fascinated because I think we are going after novel drug targets. However, it's more like we've rediscovered, because these targets or these phenomena that we're targeting, you can read about them since the early days of pathology. In fact, the father of modern pathology, the German pathologist Rudolf Virchow, was describing these very things in his journal. And these things are kind of well known, but maybe not necessarily appreciated. But the point I was getting to is that the timeline, like, it's kind of irrelevant because a lot of that's already baked into whatever operations we do today. And then it's also kind of like, so I used the example of Accenture and their ability to develop a CDK inhibitor. Super awesome chemistry, like really, really cool platform. And I don't want to trivialize that by any means. But the point that I was trying to make is that the reason why that was possible was because there was already an abundance of literature that they can work off of, that they can build off of. They can't really necessarily apply that same technology to something that we don't really know about, right? Like, there's no literature on it. So you can't feed anything to your algorithms, right? And then, there's a problem that, well, you may be able to come up with an awesome CDK inhibitor, but we still don't know if CDK7 inhibition is really going to be meaningful in the clinic. And so those are some of the inherent limitations of applying this technology in tech bio. And most tech bio companies today are going after known biology. And some of that is probably strategic, right? Because it allows you to do some benchmarking. If you go after known biology, it's harder to do that if you're going after completely novel biology, or if you're going after a disease indication where there's no other drug on the market. It's hard to benchmark, right? So that's probably one of the reasons. And the other is that, they may be deemed like a safer market because there's some validation of it. So there's probably mostly strategic reasons behind it. But the idea that it takes 17 years to develop a drug today, I don't really see it as necessarily true. Though technically you can make the argument that if you trace the history of a drug that got approved to its initial beginnings, then yeah, probably, but that's irrelevant for us in our present reality. And so I think that answered the first part of your question. There was the second part, right? So you're asking about the timelines and something else.
And then I was asking, why is it that TechBio isn't necessarily making this process faster or more efficient or just better overall?
Oh, got it. Yeah, yeah. So so to that point, the reason why, and this kind of dates back or relates back to Eroom's law, like what was so brilliant about Jack Scannell's observation is that technology has not made us more efficient, right? And so the reason why drug discovery today is getting more expensive at the same time that operations are getting cheaper, right? Because technology has made things cheaper, like sequencing genomes is super cheap compared to how it was before. It means that we're basically like that difference, that delta in what the price was before to what it is today. We're filling that up and overshooting in terms of the number of experiments that we're now running. And so we're doing more science than ever. That's one of the reasons why things are more expensive. We're doing more science than ever, and yet we're finding less things than I think it's time for us to reassess our assumptions, guys. Like maybe we've just been working off of the wrong assumptions. Like we got to be a little self-critical here. Like, it's amazing to see this in cancer biology. Like if 95% of all trials fail, then why do we keep relying on the same readouts preclinically, right? Every single clinical trial that we run first had to look amazing preclinically, right? I mean, it's hard to convince anyone to let you go into the clinic and to fund your clinical trial if your data looks like crap. So there had to be a compelling enough reason to think that you should go into the clinic. So clearly, our preclinical readouts have no relevance whatsoever to the clinic. Now, does that mean that our models are incorrect or the things that we're measuring are incorrect? My stance is that the things that we're measuring are incorrect. The things that we're looking for are incorrect. Viability assays in cancer is a perfect example. What the hell does that mean? You'll get different responses from cancer biologists if you ask them. Some people will say it's a measure of cell death. It's not. Some people will say it's a measure of robustness. What the hell does that mean? Super general. I don't know what that means. Some people will say it's a measure of proliferation, which it can be. But you can have two identical viability readouts that mean completely different things. So you can have one that you see a 50% reduction. You have two scenarios, 50% reduction in viability. One case is that you didn't kill a single cancer cell, they just stopped growing. And since you're comparing it to the control that was not exposed to the drug, basically it's normalized, right? So your control kept growing, this one didn't. So the delta, that's that 50% reduction in viability, but you didn't kill a single cell. So what the hell does it mean to say you reduced it by 50%? Alternatively, you can have a scenario where you killed 50% of the cells, and then the 50% that remained proliferated. So they made up for the cells that died. So you kind of end up with the same number of cells. Again, 50% reduction compared to the control. Those are two vastly different scenarios, clinically speaking, and from a mechanism of action. But our readouts would suggest that they're exactly the same. And that's part of the problem. So, I kind of, it's a recurring theme. Like our readouts suck and we don't really think about them, but they suck, and it's just, I get really excited and worked up about this stuff. But going back to why it doesn't increase speeds is because we're just doing more of the same faster, but we end up in the same place, which is failure. And as long as we keep failing, we're not going to improve anything, right? You can make, drug discovery 1,000 times faster and you can move 1,000 molecules more into the clinic. But if they're going to continue to fail, then we're not really doing anything. The only way to improve efficiency is by improving the success rate. You have to improve in a very meaningful way the probability of clinical success. That's the only way you're going to improve efficiency. The only way you're going to retain market share is through best in class. So my personal belief is that the only two things that matter for AI drug discovery companies are a substantially greater probability of success, because we should put ourselves to that bar, right? If we really think our technology is as great as it is, and I do think it's as great as it is, despite the bullshit that's out there, our bar should be that we can meaningfully make a difference in probability of success. And at the same time, that we can also find the best possible candidate for the clinic, right? Because you don't need an AI to get a drug approval today, right? You can do that without AI. We've been doing that without AI. So clearly, if AI is such a big difference maker, the difference should be that we're now finding absolutely the best drugs to put into the market, and that we're doing so at a much higher probability of success.
As you're talking, I'm getting this image of a sailboat, which are our traditional methods, and then a speedboat, which is AI drug discovery. And they're just both headed, the wrong heading, wrong direction. And that's kind of what's happening, right? And you're saying that we need to sort the heading out first and pick some better outcome measures, perhaps. And that will be what helps.
Yeah, exactly. That's a great analogy. But by similar analogy, some people have sort of appropriated the tech culture, and that in my opinion, and Tony from Pillar VC made this point, which I very much agree with. Tech bio is just biotech. It really is. It's the same thing. The reason why I went with tech bio is because when you say tech bio, at least you know exactly what type of phenotype of company we're referring to, which is more of like the biotech equivalent of tech, which is like younger founders, like not the old guys, like the old boys club of yesteryear. Like, not that is what it is today, but yeah, that's kind of the contrast in terms of founder phenotypes and things like that. Some people in tech bio have also appropriated the expression of like fail faster, right? Which in tech makes total sense. Like, you want to iterate as quickly as possible so you can arrive to the solution. But biology is a vastly different thing. I mean, vastly different enterprise. You can fail as fast as you want, as many times as you want. Failing does not necessarily mean that you're going to figure out how to do things. It just means that you're going to keep failing, right? I'm not interested in failing faster. I'm interested in being right, as fast as possible, as early as possible. I don't want to fail 1,000 times before I get to the right answer. I want to get to the right answer within 10 tries, That's my personal thing. I don't want to fail faster. I want to be right.
In the tech bio space, my understanding is that there's loosely three different funding models. There's the traditional public grants, there's this new kind of VC-backed companies, and then there's traditional big pharma. And there may be more. But I just wanted to get your thoughts on that landscape and where you see perhaps the pros and cons. And maybe if in those models, do you see any limitations on what we're currently able to do? And do you see any better models that could develop, essentially? So I know it's a very broad question, but if you just have any thoughts on funding and how it could be done better, I'd be really interested.
Yeah, I definitely have thoughts. As you can tell, I love to pontificate. But I think in a past life, I probably was a philosopher. I just enjoyed sitting on a rock and think. But I love thinking about things like this. But I think there's one problem that underlies all funding mechanisms, and that's groupthink, which is inevitable. It's just an inevitability of human nature, right? So first of all, like the worst thing we can do for innovation is to institutionalize things. We institutionalize public funding through the NIH, right? And it makes sense. You try to disperse that money in a productive and responsible way, and people like public figures, government bodies, want to make sure that that's what happens. But you end up adding a bunch of bureaucracy, and then you end up adding a bunch of gatekeepers, right? Because who reviews the grants? Who determines what gets funded? The people that you put in place. And what are they going to like to fund? The things that they like, right? The things that support their hypotheses. The same thing happens in VC, by the way. I mean, I appreciate the fact that VCs in some ways are risk takers, but actually VCs are incredibly risk averse, super risk averse. They may be in different ways though, but at the end of the day, they also don't like taking risks, which is also, I think, the best VCs understand the value of risk, right? There's some VCs that, are just, everyone knows about them. Sometimes, some people view them as controversial. I'm kind of indifferent, but I love, just listening to smart people. And sometimes you can dislike someone, or you may not agree with the precision. Doesn't change the fact that they're super smart people, right? And they're saying something that you should be paying attention to. So, for example, one investor is Andreessen Horowitz, and he talks about how, a lot of times, like, the best investors will optimize for those huge swings, right? Because it's a power law, the law of outliers. And that if you just get that one win, that will make up for every other loss. But what's key that he says is that VCs are remembered not by the losses or which companies that they invested in failed. They're remembered by that one company that they invested in that just made it big, right? Every investor in Facebook has been solidified in the Hall of Fame investors, right? And Ubers and stuff like that. They probably invested in 1,000 companies that failed before Facebook and after Facebook, and nobody cares because they just have that, I invested in Facebook, and automatically like, damn, you invested in Facebook. It's like elite status right there. And then the other thing, another investor that I think has a very keen insight here. He's probably a little bit more controversial. Yeah, again, I'm kind of indifferent to these things, but Peter Thiel, right? So he says that what's key to successful business and like market dominance is trade secrets or secret knowledge, essentially. I very much agree with that. Yeah, and in biotech, I think we give too much value to patents. Patents are, I mean, yeah, they're kind of necessary, but they don't give you the market protection that you think they do. I mean, there's a million different ways to target that you're going after. So it doesn't prohibit people from coming into your market and taking a market share. And it also doesn't technically prohibit people from like blatantly ripping off your product, right? I mean, this is a hypothetical. I'm not accusing anyone or any governments of doing this, but let's just say that, tensions are high between the US and China. And suddenly you realize that there's a company in China that is literally selling your drug, like they reverse engineered it, figured out the formulation, and are selling that very drug under their own name as if that company produced it, right? And so you get angry and you call upon like international patent laws and you get the government to call China and be like, hey, you guys got to shut those guys down. Maybe China isn't that incentivized to help you because they're like, well, look, we could let this company provide this drug to our people, our population for a significantly cheaper price, or we can rush to defend the interests of another government and another company so that they can sell that same drug to our people for a significantly greater price. My sense is that they probably wouldn't be that interested in helping out. They might be like, yeah, we'll look into it eventually, maybe. Yeah. And so you don't really and that's a small company. I mean, good luck trying to get, to enforce some of these policies, right? So they can your IP is easily stolen as well. So anyways, the trade secrets, I think are a big difference here. So about funding models, going back to funding models, the problem I see with VCs a lot is that group thing. I see it in TechBio. And I kind of call some of those things out where everyone starts writing thought pieces and then they all start agreeing with each other. And then they all sort of assimilate the same information, the same ideas. And then they collectively decide that this is what we're going to fund. So if you get someone that's outside of those schemes, they may be very right, but they're not within those schemes, right? And so, but yeah, I'm not necessarily blaming them, right? So I mentioned earlier that you have to understand human nature. It's essential. It's indispensable. And think of it from the investor standpoint. They get inundated with deal flow that you just don't have the mental bandwidth to even I can't imagine looking through decks after a while, they would all look the same to me. You don't have the energy probably to really think critically about what they're trying to tell you. So what do? You start deferring to social signals, right? And then there's also an inherent fear of looking stupid, right? Any human has that you're like, well, what are my buddies investing in, right? And so you may like this something, but you don't really know the team. You just, you think there's something there and you kind of liking it. But then one of your buddy investors comes in, he's like, no, forget about those guys. I got my champion right here for us. Let's syndicate around this guy and let's invest in this guy. Screw that other team. No, this is our guy. I've known this guy. We went to Stanford together, blah, blah, blah. And that's exactly how VC syndication works. And so you have to understand social signals. You have to understand the decision-making process. So at the end of the day, you still get groupthink in VCs. But what I like more about the VC model than the grant model is the efficiency of it. You can get from a no to a no, or from a no to a yes. You get from a no to a no a lot faster, but nevertheless, you can usually get to that within a few weeks to a few months, right? I mean, if it's taking more than 2 months, either you're doing a super large raise that just requires much more level of due diligence, but for the most part, you can learn as quickly as a week. Whereas the grant model can take literally months before you even know if you got the grant, and then many months more for that grant money to come in. And then some of that you have to pay automatically to the university. There's that overhead fee. So if you're wondering why American universities love research, it's because from every grant that their scientists get, you get a chunk of it. It's a huge revenue model. And so, at the end of the day, the problem is always human. We're the thing that corrupts everything.
The last thing I wanted to ask you about was, have there been any habits or ways you like to approach problems that have been helpful for you in your career and along your journey?
Yeah, for sure. I mean, I think this is probably more of a trait than a habit. I think I'm a very ADD person and I'm a very curious person. So naturally, for example, for me, I love science because it's just endless rabbit holes. It's the best thing I can ask for. I'll just get lost in those rabbit holes. I love it. But, when we enter a new, domain area or, a new field or something, we want to kind of learn about it, right? Usually what we do is we'll try to figure out very quickly, like, what is the consensus, right? What is everyone saying? What is the state of the industry, or the state of knowledge for this particular field, et cetera, et cetera. So like if I want to learn about immunology and start reading a bunch of immunology reviews or whatever, what we typically do is then build off of that. But in my case, I'm just kind of like, very curious, and I have an insatiable curiosity where I start, instead of building up from that, I start digging in. I'm like, well, okay, so this is a consensus, but why are we saying this? Now, I always joke that my superpower is asking all the stupid questions. And, people say there's no such thing as a stupid question. Oh, there is. I've asked plenty of them. Trust me. What there's every now and then, what is seemingly a stupid question is actually a quite brilliant question, because oftentimes most impactful things are things that were sort of in plain sight, but no one ever really saw them. Right. So I gave you an example of cancer and the definition of cancer, like dormancy. Everyone knows about cancer dormancy, but no one makes a connection of like, wait, that's a sharp contrast to the idea that cancer is a disease of uncontrolled proliferation. Clearly it's controllable, right? Something controls it. I like to dig in and really understand why we say the things that we say. And I consider myself a very slow learner. But I think the reason is it's part of that ADHD or ADD, where it's not so much that I learn slow. It's just that I'm processing more things at the same time, because I think like attention deficit is such a big misnomer. It's the opposite. You're paying attention to too many damn things at once. It's more of a focus, a deficit, right? And so my filter, normally like you're like, I want to learn this. And so you have a pretty like refined filter for that. And that's good from an efficiency standpoint. My filter is like this. I'm thinking about a lot of things at once. I'm a slow reader, I think, because of that, because I'm really like this. My wheels are churning as I'm reading. And so it takes me a while to learn something. But I feel like once I do learn something, I have a pretty solid grasp, because I've contemplated all these different like weird tangents that may be seemingly unrelated. But then in my mind, I'm like, oh, that kind of reminds me of this. And so, yeah, it's like a creative web of ideas in my mind. And so, maybe some of that is a character or trait of mine. But I would say that I think I found it incredibly helpful for me as a professional, and especially as a scientist, to take that level of curiosity and apply it to everything that I'm learning, and to really like ask the dumb questions like, okay, rather than working off of assumptions, why are these assumptions even built in? Because most of the time we're working off of built-in assumptions that we don't even realize. And I think that is, if you can, that's where kind of alluding back to what Peter Thiel said, like those trade secrets. Like suddenly you learn something that may seem trivial, or maybe it was so obvious, as they say, it's hidden in plain sight. But that completely transforms the way you go about doing things.
I hope you enjoyed that episode. You can find all my links by going to bigpicturemedicine.co.uk. And if you've been enjoying the podcast, then please consider leaving a review. By the way, all of these episodes are now available on Spotify and on YouTube in video format. Thanks for listening.